Linköping University, Sweden, March 27, 2020. A genomics paper lands in Scientific Reports. It's not a splashy paper. It wasn't announced with a press release. The stated purpose was pharmacogenetics, figuring out how Somali patients metabolize medication differently than European patients. Practical, medical, quiet. But buried inside the data was a principal component analysis plot, a map that shows how populations relate to each other genetically, and something on that map stopped the analysis cold. 95 Somali individuals from Puntland, northeastern Somalia, mapped against every neighboring population on Earth. And when the plot rendered, the Somali cluster didn't land near Yemenis, it didn't land near Egyptians or Sudanese, it landed, with geometric precision, right next to Ethiopian Jews. Nobody sent out a press release about that either. Now, here's the consensus answer to why that happened. Scientists have known for 20 years that Somalis carry approximately 40% West Eurasian genetic ancestry. Jewish diaspora populations, from Ashkenazi Jews in Eastern Europe to Mizrahi Jews in Iraq, also carry strong Levantine ancestry. Two populations with overlapping Middle Eastern DNA profiles cluster near each other on a genetic map. Comparison made, similarity confirmed, story filed. Every number in that explanation is accurate. Every single one. And every single one of them is pointed in the wrong direction. Stay with me, because this next part is the reason this video exists. The primary haplogroup carrying that so-called Levantine ancestry in Somali men, a marker called E1B1B, found in roughly 70 to 77% of Somali males, does not have its origin in the Levant. It has its origin in Africa. It migrated into the Levant approximately 15,000 years ago. Not from the Levant into Africa, from Africa into the Levant. The direction that every textbook, every ancestry website, every comparison study has been running, that's the direction it ran after the original event. The original event ran the other way. Abshir A. Ali and his team at East Africa University in Bosaso, Puntland, were not looking for this. Their 2020 study in Scientific Reports examined nearly 900,000 genomic markers across 95 individuals. The finding that Somalis cluster near Cushitic and Semitic-speaking Ethiopian populations, including Ethiopian Jews, appears in the results section. It is noted, it is not pursued. Because when your paper is about drug metabolism and immune markers, you note the unexpected clustering and you move on. Nobody came back. If you've ever genuinely wondered where your ancestry actually begins, not the family story, not the country on the passport, but the biological record that has been running in human cells for 40,000 years, subscribe. Because what we're about to trace goes back further than any ancestry test has ever told you, and it runs in a direction nobody has been advertising. The story everybody knows says that Somalis and Jewish populations share a piece of Middle Eastern ancestry. That part is true. What that story leaves out is what happened before the Middle East was a genetic source, before it was the point of origin, before it had anything to give. And the answer to that? Nobody published a correction. The correction was never published because the question was never formally asked. What was published repeatedly across 2 decades of population genetic studies was the observation: E1B1B appears in Somalis, in Berbers, in Ethiopians, in Jewish populations, in Greeks, in Italians. The distribution was mapped. The researchers who specialized in haplogroup origin chronology noted the African source, and that finding lived in its own corner of the literature. The researchers doing population comparisons used the distribution data. The two bodies of work never talked to each other. That gap has a name. It's called not reading across subfields. It's extremely common, and in this case, it produced a direction error that has been structuring how we understand the relationship between East Africa and the Middle East for 20 years. Now let me show you where this started. Rockafet Cave, Mount Carmel, northern Israel. Approximately 12,000 BCE. A burial. The archaeobotanists who excavated the site found impressions in the consolidated sediment. Impressions left by flowers that had been scattered in the grave. These are the Natufians, the first sedentary culture in the Levant. They didn't wander, they stayed. They built. They buried their dead with flowers. And when researchers extracted Y-chromosome data from 6 Natufian male remains at this site, they found haplogroup EM215. the direct parent clade of E1b1b, the haplogroup that defines Somali male lineage, in a 12,000-year-old grave in northern Israel. But here's where it gets strange. In 2018, Daniel Schreiner at the National Human Genome Research Institute took a fresh look at Natufian genetic samples, this time running them against 279 modern reference populations. What he found was that the Natufians were largely of West Eurasian origin, yes, but they also carried 6.8% Eastern African-related ancestry, specifically an Omotic component, the kind that peaks today among Omotic-speaking groups in southern Ethiopia. And his team's proposed mechanism, that haplogroup EM215, E1b1b, was introduced into the Levant from East Africa. Not born in the Levant, arrived there from the south. The Natufians didn't originate E1b1b, they received it. And then they became one of the founding populations of the Levant, which means the most critical genetic bridge between East Africa and the ancient Near East was built by a population carrying an African lineage that had already moved north. Here's the part that changes everything. The Natufian individual buried with flowers at Rakafet Cave carries a paternal lineage whose highest frequency modern home is not Israel, not Lebanon, not any country in the Middle East at all. The highest modern frequencies of E1b1b are in Somalia, Ethiopia, and Egypt, in that order. The ancestor of both Somali and Jewish genetic ancestry is lying in a cave in northern Israel. He has been there for 14,000 years, and no modern population has claimed him. No ancestry company has a category for him. No flag represents him. He has no name in the present tense. The separation happened in two places at the same time, and that's the part that nobody has explained. In the Horn of Africa, around 3,700 years before present, a genetic bottleneck consolidated the Somali male population around a specific deep subclade of E1b1b, a variant called EV32, and more precisely EY18629. The formation date, 3,700 years before present. The time to most recent common ancestor of the majority of living Somali men, 3,300 years before present. That's not ancient history in the way people usually mean it. That's the Bronze Age. Now, same era, the Levant. Bronze Age population formation events are crystallizing the ancestral populations of what will become, over the following 3,000 years, the world's Jewish diaspora communities. Two biological clocks, one in the Horn of Africa, one in the southern Levant, both ticking toward the same interval. That number, 3,700 years before present, stopped me cold when I found it. Mary Prendergast at St. Louis University and Mark Lipson and David Reich at Harvard Medical School published a study in Science in 2019 that mapped the genetic history of the earliest herders to enter sub-Saharan Africa. They extracted genome-wide data from 41 individuals buried at pastoral Neolithic sites in what are now Kenya and Tanzania, people who lived there between roughly 5,000 and 1,000 years ago. What they found was this. These ancient herders carried ancestry from two distinct sources, roughly half Nilotic-related ancestry and roughly half ancestry related to pre-pottery Neolithic Levantine farmers. The same Levantine source that feeds into the ancestral Jewish population formation story. And among modern populations, the closest genetic match to these ancient herders was not any modern Middle Eastern population. It was modern Cushitic speakers, Somalis, Oromos, Afars. This discovery was recorded in 2019. It didn't make the comparison studies. Here's why this matters, and I want you to hold the numbers for a moment. The deep subclade that defines the majority of living Somali men consolidated around 3,700 years before present. The pastoral Neolithic herders, who are the closest ancient analog to the ancestral Cushitic population, carried Levantine ancestry from the same period. And the Bronze Age Levantine populations that seed the ancestral Jewish diaspora were forming at the same time. 3 processes, 3 geographies, the same century. In geology, we would call that a tectonic event. Multiple fault lines moving simultaneously in response to the same deep earth pressure. In genomics, the field has no term for it. The closest word in the literature is coincidence. The word coincidence has never been formally challenged. The most significant challenge to the received narrative was published in 2010. Not quietly, it landed in Nature, one of the most comprehensive genome-wide studies of Jewish populations ever conducted. Doron Bahar and a large international team analyzed 14 Jewish diaspora communities and compared them against 69 non-Jewish populations from across the Old World. The results confirmed the dominant finding that most researchers had been building toward. The majority of Jewish diaspora communities trace a significant portion of their ancestry to the ancient Levant. That finding was real, it was important, it held up. But the study contained one result that the abstract's first sentence did not mention. Ethiopian Jews, the Beta Israel community, did not cluster with Ashkenazi Jews. They did not cluster with Sephardi or Mizrahi Jews. They clustered with their Ethiopian neighbors. And those Ethiopian neighbors, when placed on the same genomic map as Somalis, as Ali et al. Would show 10 years later, sit at the same genetic address. And yet, the data said the exact opposite of what the headline implied. The headline said most Jewish communities trace to the Levant. True. The data also showed that one Jewish community, one with thousands of years of documented, living, practiced Jewish tradition, traces genetically to Cushitic East Africa. And that Cushitic East African genetic profile, when you map it against Somali genomes, sits in the same neighborhood. A principal component analysis plot is a powerful instrument. It shows you where populations land relative to each other. What it cannot show you is the ancestor they both started from before they separated. It can show you the distance between 2 points. It cannot tell you what stood at the origin. The Beta Israel practiced Judaism in Ge'ez, an ancient Ethiopian Semitic liturgical language. Their Y-chromosome haplogroup distribution is predominantly East African. A 2012 study by Harry Oster at Albert Einstein College of Medicine and Karl Skorecki at the Technion Israel Institute of Technology confirmed that while Beta Israel more closely resemble indigenous Ethiopian populations, they carry detectable paternal ancestry tracing back to the Levant, approximately 2,000 years distant. They exist at a biological coordinate that the PCA plot was not designed to explain. And here's the part the literature hasn't reckoned with. The Beta Israel are not a genetic anomaly in need of explanation. They may be the most accurate surviving biological signal of what the ancestral population looked like before the Jewish diaspora and the Cushitic expansion ran in opposite directions from the same starting point. The categories, Jewish, Cushitic, Somali, Ethiopian, are newer than the genomes. The genomes don't care about the categories, they just carry the signal. Actually, hold on for a second. Before we go further, I want to ask you something directly. If you've taken a commercial DNA ancestry test, did any of your results surprise you? Not what you expected to find, what you didn't expect the test to miss. Drop it in the comments, I read them. Because what this research is telling us is that many of those tests are showing you the right numbers with the wrong labels. And what you think was missed might not have been missed at all. It might have just been called something else. Something geographically convenient. Something that pointed in the wrong direction. Djibouti City, present day. Population approximately 600,000. A port city on the Gulf of Tajura, where the Horn of Africa narrows to within 30 kilometers of the Arabian Peninsula. Among the Somali deer clan members documented in Djibouti, researchers found the following when testing paternal Y-chromosome haplogroups. 100%, haplogroup T. Not a high frequency, not a majority. Every single sampled deer male, the entire group, a West Asian lineage, saturating a Cushitic East African population at a rate that doesn't exist anywhere in West Asia itself. Not in Yemen, not in the Levant, not in Arabia. In Djibouti, that number stopped me cold. Haplogroup T, formerly TM184, is the second most common Y-chromosome lineage in Somali men overall, at approximately 11%. That alone is notable because haplogroup T is also found at frequencies ranging from 15 to 30% across multiple Jewish diaspora communities globally. Ashkenazi, Sephardi, Mizrahi, it appears in all of them. That overlap had been noted, that comparison had been filed, but the deer frequency, 100% in Djibouti, had not been placed at the center of the conversation where it belongs. Here's the part that changes everything. Researchers calculated the TMRCA, the time to most recent common ancestor, for Somali haplogroup T lineages. The result? approximately 2100 to 2200 years before present. That is not the Neolithic, that is not the Bronze Age, that is 150 BCE, the Hellenistic period, the century of Alexandria, the century of the Maccabean Revolt, the century when a specific body of water, the Bab el-Mandeb, the strait connecting the Red Sea to the Gulf of Aden, was one of the most trafficked trade passages in the known world. Something crossed that strait or moved along its coast between approximately 200 and 100 BCE, something that carried haplogroup T at high concentration and consolidated it in the space of a few generations into the paternal lineage of the deer at a frequency that no population in the Middle East has ever matched. Among global Jewish populations, haplogroup T peaks at around 30%. The deer of Djibouti, a Cushitic-speaking Muslim community on the East African coast, hold it at 100. No one has identified the population responsible for that event. Their descendants walk through Djibouti today, carrying a Y-chromosome lineage that ties them, at frequencies higher than anywhere in the Middle East, to a genetic signature shared with Jewish communities worldwide. And the event that produced it has no name, no recorded history, and no archaeological trace beyond the frequency itself. A frequency that precise is not an accident. That is a memory, written in DNA instead of text. I want to show you two things at once, two stories happening simultaneously in different geographies. They do not connect, they do not interact, but they are the same event. The Rift Valley, Kenya, approximately 3000 BCE. A group of herders move south from northeastern Africa. They carry cattle, the first domestic cattle to reach this region. They carry a genetic profile, roughly half ancestry related to Levantine Neolithic farmers, roughly half ancestry related to Nilotic peoples. They settle near Lake Turkana. They build nothing that history will name. They leave their bones in the earth. And 4,900 years later, a research team at Harvard extracts DNA from those bones and finds that these people were, by every genomic measure, the closest ancient analog to modern Cushitic speakers, to Somalis. At the same time, the Judean hills, Canaan. Approximately 3000 BCE, agricultural villages are forming across the Levant. Their inhabitants carry an ancestry that is predominantly Natufian descended, mixed with populations from the Zagros Mountains and the Caucasus. They are building nothing that will be called Israel for another 2,000 years. They leave their bones at Megiddo, at Hazor, at Ain Ghazal. And in 2025, an international research team including Israel Finkelstein's group at Tel Aviv University published a Bronze Age ancient DNA study confirming that modern Jewish populations share more than 50% of their ancestry with these people, more than half, from the Bronze Age. These are not the same event. They are not connected events. They share no direct historical relationship. Everything I just told you, hold on to it, because it's about to mean something completely different. Because both groups, the pastoral Neolithic herders in Kenya and the Bronze Age Levantine villagers in Canaan, derived their defining ancestral component from the same source. Prendergast and Lipson and Reich's 2019 study confirmed it. The Levantine ancestry in the pastoral Neolithic herders came from populations related to pre-pottery Neolithic Levantine farmers. The same pre-pottery Neolithic Levantine population that the 2025 Bronze Age study confirmed as the primary ancestral source for modern Jewish communities. One population, seeding both lines simultaneously. Two shores, one tide, moving in opposite directions at the same moment. One southward into the Horn of Africa, building what will become the Cushitic world. One westward and northward into the Mediterranean basin, building what will become the Levantine and eventually the Jewish diaspora world. They will not encounter each other for 5,000 years. When they do, a genomics lab in Sweden will map their descendants on the same plot. The clusters will land near each other. The researchers will describe this result as expected. The researchers will call this finding expected. Take a moment with that word, expected, as though the convergence of two populations separated by 5,000 years and 3,000 kilometers, both descending from the same pre-pottery Neolithic ancestor, was simply the predictable output of a well-understood model, as though naming it expected was the end of the question rather than the beginning of a very different one. If that ancestor had not crossed the Turkana Basin into East Africa around 3000 BCE, The Cushitic genetic profile does not form. The modern Somali population, as a genetic category, does not exist. 30 million people are alive today because one group of herders crossed one threshold 5,000 years ago. That isn't expected. That is the narrowest margin in human history. A quick note before we hit what I think is the most important part of this video. In our last video, we traced how a population's genetic record preserves the memory of a migration that official history never named, how the science arrives before the story does. And right here, in this exact moment, we're watching it happen again from the other side. Two populations descending from the same ancestor. The science records it. The framework hasn't caught up. If you watched that last video, did you notice the same pattern running through both stories? Drop it in the comments. Because what's coming next in this episode is the moment that breaks the framework entirely. What Ali and his colleagues at East Africa University did not pursue, what nobody came back to pursue, was the specific implication of where the Somali cluster landed on that PCA plot, adjacent to Ethiopian Jews. The paper was about pharmacogenetics. The clustering result was a note in a results section, and the study moved on. Then there's the Genetic Atlas of Human Admixture History, a 2014 study in Science that used genotype data from nearly 1,500 individuals across 95 populations to map admixture events across 4,000 years of human history. East Africa appears. The Levant appears. Their shared ancestral component is identified, mapped, measured, and labeled. The label the field gave it was Levantine ancestry. The arrow on every map, in every study that built on this atlas, points west to east, from the Levant into Africa. This is the moment. Stay with me. This isn't just ancient history. This is in your DNA right now. E1b1b, the defining paternal lineage of Somali men, and a significant marker in Jewish male populations globally, does not have its origin in the Levant. The oldest confidently placed samples of this haplogroup's parent lineage come from Northeast Africa. When Daniel Schreiner analyzed the Natufian samples in 2018, his proposed mechanism was explicit. EM215 was introduced into the Levant from East Africa. The Natufians received it. They did not originate it. What this means, and I want to say this as precisely as possible because precision is everything here, is that the genetic element most commonly cited as shared Levantine ancestry between Somali and Jewish populations is, at its oldest traceable layer, shared African ancestry that moved into the Levant 15,000 years ago, became part of the Natufian founding population, was transmitted through the pre-pottery Neolithic Levantine farmers, who then seeded both the Cushitic East African herder tradition and the Bronze Age Levantine village tradition, and emerged 5,000 years later in two populations that comparative genomics then described as sharing Middle Eastern DNA. They do share Middle Eastern DNA. That part is accurate. What they share underneath it is African DNA that the Middle East received first. Now, this does not diminish the Levant. It does not reduce the genuine, confirmed, ancient DNA-verified Levantine ancestry that Jewish populations carry. Ancestry that Bronze Age genomics has now confirmed with high statistical confidence. That ancestry is real. Those people were there. That history happened. What it means is that inside the Levantine signal, before the Levant was a genetic source, before it had anything to transmit, is a layer from Africa. A layer that the naming system of population genetics labeled by the place it passed through, not the place it came from. And here's the finding that makes this biological, not just historical. The HLA immune architecture of the Somali population, the body's own system for recognizing and remembering pathogens, was found in the Ali et al. Study to diverge from every neighboring population tested. Ethiopians, Kenyans, Sudanese, Arabs, every neighbor, except one cluster, the Ethiopian Jews. HLA is not a haplogroup. It's not a migration signal. It's immune memory. It records what diseases a population survived. And if two populations share an unusual HLA profile that sets them apart from all their geographic neighbors, that is the genome saying, in the language of biological survival, we went through something together, something specific, something that left a mark. What that event was, the data has not told us yet, but it happened. It left a record. And that record is sitting in a 2020 study about drug metabolism that nobody built a press release around. Bosaso, Puntland, Somalia, 2018. A researcher at East Africa University begins collecting saliva samples. 95 individuals, unrelated, consenting, from across northeastern Somalia. The study's stated goal: to understand how Somali patients respond to common medications. Pharmacogenetics. There was no grant for ancestry research, no press outreach planned. Just a clinical question about drug safety in an understudied population. 2 years later, those 95 samples become 95 data points on a genomic map. And one of those data points is the result that placed the Somali cluster next to the Ethiopian Jews. Not because anyone was looking for it, because the data put it there. The HLA findings in that study are the part that has stayed with me longest. HLA, human leukocyte antigens, is the immune system's recognition architecture. It's the part of your genome that decides how your body identifies a pathogen and builds a response against it. And crucially, it is heritable. You carry the immune memory of what your ancestors survived. What Ali and his colleagues found was that the Somali HLA profile didn't look like any of its neighbors. Not Ethiopians, not Kenyans, not Yemenis or Egyptians. It looked like the cluster occupied by Cushitic and Semitic-speaking Ethiopian populations, including, at its closest point, the Beta Israel. There are 3 things that finding could mean. It could mean shared recent ancestry, populations mixing in historical times. It could mean shared ancient ancestry, a common source population whose immune adaptations both groups preserved. Or it could mean something more specific, that at some point in the deep past, the same epidemic, the same pathogen, the same crisis swept through a population that was the common ancestor of both communities, and both groups carry the immune response their shared forebears built to survive it. The data doesn't tell us which, but consider what the Haplogroup and PCA evidence already established. Natufian origin ancestry feeding both Cushitic East African and Levantine population streams. The same ancient genetic source. Now add an immune architecture so similar between Somalis and Ethiopian Jews that it separates both groups from all their geographic neighbors. That is not one coincidence. That is a pattern. Now, here is where the story should resolve. The HLA data, the haplogroup data, the PCA clustering. Taken together, they build a coherent picture. Somalis and Ethiopian Jews share a pre-linguistic, pre-religious, biologically recorded common ancestry that is deeper than either group's named identity. The genomes agree, the immune systems agree, the paternal lineages agree. And then the final piece of evidence arrives, not a finding, an absence. The entire archaeogenomic case for Somali population history, every haplogroup frequency, every PCA plot, every HLA cluster we've discussed in this video is built on living individuals. Not ancient DNA, not skeletal remains, not bone fragments from a burial site, living people. Because ancient DNA from Somalia, from the peninsula that has been inhabited by anatomically modern humans for at least 100,000 years, has never been successfully extracted and published. In the Allen Ancient DNA Resource, the most comprehensive publicly available ancient genome database maintained by Harvard and the Max Planck Institute, the Horn of Africa is represented by one ancient Ethiopian individual, the Mota, from approximately 4,500 years before present. Somalia in the database is white space. Everything we know about Somali genetic ancestry is a reflection in living mirrors. And living mirrors can only show you the present. They cannot show you what stood at the beginning. Open the Allen Ancient DNA Resource, version 54 and later. The most comprehensive catalog of ancient human genomes ever assembled. Thousands of individuals, Neolithic farmers from Anatolia, Bronze Age warriors from the Eurasian steppe, Mesolithic foragers from Portugal, Iron Age merchants from the Levant, ancient Egyptians, pastoral Neolithic herders from Kenya and Tanzania, the same ones whose genomes told us about the shared Levantine ancestry feeding into Cushitic origins. All of them in the database. Somalia, 0. Not sparse, not underrepresented. 0 published ancient genomes from the Somali Peninsula. This isn't a criticism of anyone. Ancient DNA extraction is expensive, logistically complex, and politically sensitive. Somalia's recent history has not made fieldwork easy. The absence has reasons, but the absence is real, and it matters enormously for everything we've discussed in this video. Because every comparison we've made, every cluster we've identified, every HLA overlap we've noted, rests entirely on living populations as proxies for their ancient ancestors. And proxies are not the same as evidence. Here is what that means for you specifically, and I mean you, the person watching this. If you carry ancestry from East Africa, the Horn, the Arabian Peninsula, the Levant, North Africa, or any Mediterranean population, you carry a component of the signal this video has been tracking, not as a metaphor, literally. The haplogroup origin data, the pastoral Neolithic ancestry proportions, the HLA immune architecture, these are biological structures inside your cells that have been running since before any of the place names we've been using in this video existed. The Horn of Africa, the Levant, the Middle East. These are modern geographic labels on prehistoric human biology. The biology doesn't know those names, it just carries the signal. What we have called shared Levantine ancestry between Somali and Jewish populations contains, at its deepest stratum, an African layer that was renamed by the place it passed through. What we have called an unexpected PCA clustering was predictable the moment someone asked where E1B1B came from instead of just where it went. What we have called a medical finding about drug metabolism contains, in its results section, a genomic proximity between two communities that 5,000 years of separate history, separate language, separate faith, separate geography has never been fully reckoned with. If this gap in the record concerns you, if that white space on the ancient DNA map of Somalia feels like a question that demands an answer, subscribe. Every episode in this series follows one methodology: what the data contains, what the data cannot see, and the specific shape of what's missing. And in this case, the shape of what's missing is the size of 100,000 years. 30 million people carry a genome built in part by a Natufian who buried someone with flowers in a limestone cave in what is now northern Israel, 14,000 years ago. That Natufian has no country, no flag, no ancestral claim in any court or any parliament, only descendants on two continents, in two great civilizations, across two of the world's major faith traditions, who have spent centuries being compared to each other, and finding, every time the comparison is made carefully, that they share something older than any of the names they use to be different.